Hello students, I hope you all are watching my videos. So in our last class we were discussing about lymphoid organs, that is thymus, then a secondary lymphoid organs, tonsils, spleen, isn't it? So also in our last class we discussed a very important topic that was AIDS, isn't it? So what is AIDS? Which is the causative agent for the AIDS? Then what is the life cycle of AIDS? What are the symptoms? Isn't it? So we all include. In today's class, before entering into the actual chapter, I will try my best to quickly revise those previous slides. That is, AIDS means acquired immunodeficiency syndrome. I told you people in the last class, acquired immunodeficiency syndrome means, so ourselves we are going to reduce our immune system. So for that reason, many infections or diseases may attack to the individual or to the AIDS patient. Next, the causative agent is HIV, that is human immunodeficiency virus, means that virus which deficient or reduce the human immune system, so called as HIV, that is human immunodeficiency virus. That virus is come under retrovirus. I told you retrovirus means group of virus which contain their genetic material, that is single stranded RNA as their genetic material. Those all viruses are grouped into a single category, that is retrovirus, which contain single stranded RNA. Then syndrome means group of diseases, definitely group of symptoms. Next, AIDS was first reported in USA, later in India in 1986. Then more than 25 million people have died because of HIV. Then transmission of HIV is by means of sexual contact with the infected person, sharing of infected needles or blades, then transfusion of contaminated blood from donor to recipient without screening the blood, then infected mother to her respective child. Then there is always a time lag, like incubation period. So the day of entry of pathogen into the body, then appearance of symptoms. So it is usually from months to years, that is almost 5 to 10 years. Then this is what the diagrammatic representation of HIV virus. Then life cycle of HIV, isn't it? So most important, that is life cycle of HIV. That is when any HIV virus enter into the body, the first and foremost is it is going to recognize or identify specifically T lymphocyte. In that, one particular generation called as T helper cell. Clear? So with the help of that GP120 or GP40 called as glycoprotein 120 and glycoprotein 40, they simply considered as surface receptors. Next, that process is considered, step is considered as attachment. After the attachment, that particular virus release its respective genetic material into the cytoplasm of host cell called as single stranded RNA, isn't it? So when that single stranded RNA virus, a single stranded genome enters into the cell cytoplasm, then it starts undergoing transcription. Clear? In a simple way I can show you. See, imagine this is what? Imagine it is T cell, refers to T helper cell, isn't it? So this is called a cytoplasm. Within the cytoplasm, imagine it is nucleus. Clear? So within the nucleus, as we know that we will be having a double stranded DNA, isn't it? So when HIV virus enter into the body, that is, it is going to attach to the T helper cell. So which completes first step called as attachment. I hope you understand. Clear? So after the attachment, this virus which contains single stranded RNA as its genetic material. So now this virus release its genetic material or genome called as single stranded RNA. Simply it is denoted as ssRNA, means single stranded RNA. I hope you understand. Clear? So now, with the help of a process called as reverse transcriptase enzyme, called as reverse transcriptase, means the enzyme which play a very important role in the conversion of single stranded RNA to double stranded DNA. So now the single stranded RNA is converted into double stranded DNA called as viral DNA. Don't remember. Now it is called as viral DNA. So this process is called as third one, that is conversion. First one attachment, second one this is called as second step, that is release. After release, the single stranded RNA is converted into double stranded DNA, is called as conversion. Now it is considered as viral DNA. Now this viral DNA starts migrating from cytoplasm to nucleus, clear, for incorporation. It means now the viral RNA can be incorporated within the cell DNA. Now this is considered as incorporation. The step is called as incorporation. So after the incorporation, this viral DNA incorporated within the host DNA or T helper cell DNA, isn't it? So gradually the whole cell is under the control of viral DNA. It means that the whole DNA is going to become viral DNA. Do remember. Earlier it was considered as cell DNA or nuclear DNA, but now after incorporation the whole DNA is simply considered as viral DNA, so that the whole cell is under the control of each and every cell organelle. The function of each and every cell organelle is under the control of this viral DNA. Most important. Clear? Now, so this whole viral DNA starts producing. Very important. So earlier the cell DNA was able to produce the proteins, enzymes, or whatever the things which are required by the cell for its normal function, isn't it? But now the whole cell is under the control of viral DNA. Now this viral DNA is going to produce the proteins or whatever the chemicals which are required by the virus, so that this viral DNA is going to produce the viral particles. Do remember, it is going to produce the viral particles or daughter viral particles. Very important word. Clear? So once these all viral particles are going to produce within the cell cytoplasm, what happens? Now this is called as biosynthesis. This step is called as biosynthesis. So after production of viral particles, these all viral particles are going to break the cell membrane. Very important word. So they are going to break the cell membrane. The process called step called as lysis. After the lysis, these all viral particles can be released out of the cell. I hope you understand. Now these all viral particles can be released out of the cell. And now very important thing is, these all viral particles, each and every viral particles are ready to attach one more T helper cell. Clear this? So each and every cells are ready to attack new TH cells. Clear this? It means that when any TH cell is infected by the virus, at the end or eventually the destruction of T cell. Am I right or wrong? So destruction of T cell because the rupture of cell membrane. So at some extent within our body, the T helper cells, the number or the count of the TH can be reduced. Clear? So this is what? The reduction in the immunity or deficient in the immunity. Clear this? So this is what? The life cycle of HIV. So first one attachment, second one release, third one with the help of reverse transcriptase, the single strand RNA is converted into double stranded DNA called as viral DNA. This gets incorporated in the cell DNA called as incorporation. After that, the whole cell is under the control of viral DNA. Then it continues to produce the viral particles or daughter viral particles called as biosynthesis. After reaching a certain stage, all the viral particles can be break open the cell membrane and comes out of the cell called as lysis. Same time, these all new viral particles are ready to attack new T cells. So, or macrophages, resulting in the reduction or the number can be reduced. That is T helper cell in the blood. So, after when they attach, finally they are going to break. It means so, and at some extent, the T helper cells and macrophages number, their count can be reduced in the blood. Next, this is what the life cycle. First one, virus attachment. It can release the single stranded RNA. So then, single stranded RNA to double stranded DNA. Then this viral DNA incorporate in the genome, isn't it? So after that, it is going to produce the new viral particles. So these all viral particles can be break open the cell and comes out of the cell, called as lysis. Then a flowchart or like a mind map. Very simple. Try to understand this sequence. First one, the retrovirus refers to HIV virus. Virus infects normal cell. Then viral RNA is introduced. So this contain called as attachment. Then viral RNA is introduced into the host cell called as release, isn't it? So then reverse transcriptase. Reverse transcription means single strand RNA is converted into double stranded DNA called as viral DNA is produced by the reverse transcriptase called as conversion, isn't it? So then viral genetic material in nucleus of the host. So viral DNA incorporates into the genome, that is host genome, called as incorporation. Step called as incorporation. New viral RNA is produced by the reverse transcriptase. So virus comes out of that. That is, this condition is called step called as biosynthesis. This steps considered as biosynthesis. Later, viruses come out of the host cell, isn't it? So called as lysis. This step is considered as lysis. So new viruses can infect other T cells. So this is what the mind map or flowchart. Very easy to remember the whole life cycle of HIV. Very simple. Next. Then very important, due to reduction of T helper cells in the blood. Clear? So when virus infection, after that, so the T lymphocytes or T helper cells can be, their count can be reduced in the blood. So now what happens? The person becomes immune deficient. Clear? There the immune system can be reduced because of absence or a destruction of T lymphocytes. Within the individual, the count of the T lymphocyte or T helper cells can be reduced, so and become more prone to disease. Means the person may expose to the many kind of diseases or infection. They're more prone to disease means more chance to get disease. Very simple word. So where the immune deficient can be reduced means that person may get more chance to get disease. So he may easily get infection by virus, viral diseases, bacterial diseases, protozoan, fungal diseases, many. Clear? Why? Because there is no resistance power at all, isn't it? There is no resistance power at all. So for that reason, there is more chance to get disease. Next, how to diagnose the disease? So there are some specialized tests to conduct to diagnose the HIV. In that, first one is ELISA. That is enzyme linked immunosorbent assay. Please note it. ELISA refers to enzyme linked immunosorbent assay. So this is what ELISA refers to. It is antigen plus antibody reaction. So this can be understood by antigen antibody reaction. I hope you understand. So the ELISA test is, the principle behind this ELISA is antigen antibody reaction. Clear? So it means when you take the blood from the patient, that blood contains HIV virus, isn't it? So their HIV virus is considered as an antigen. So our doctors or in the laboratory, they are going to take the antibodies which are respect to this antigen. Clear? So then there is a reaction between antigen and antibody. So from that we are going to understand the person which contain antigen, that is HIV virus. So he is suffering from HIV. I hope you understand. So the principle behind the ELISA is enzyme linked immunosorbent is antigen antibody reaction. Clear? Next, one more important is that is CD4 cell count. I told you people. Clear? So the person who is suffering from HIV means that the count of T H cells can be reduced because of destruction of T helper cells. So simply called as CD4. I forgot to tell you, that is TH cell. TH refers to T helper cell. So this contain a cell receptor. This contain cell receptors called as CD4. Clear? Called as CD4. Here the word CD4 refers to cluster differentiation 4. Simple word, CD refers to cluster differentiation 4. So this is what a cell receptors. It means that CD4 refers to T helper cell. Clear? So when the CD4 cell is, the normal CD4 count cell or normal T lymphocyte or T helper cell in the blood is 500 to 1200 cells per mm cube. That is milli microcube. Clear? So in a normal person, the count of the TH cell is almost 500 to 1200 cells. That is mm cube. But in case of HIV patient, the number, the count of the CD4 or TH cell is less than 200 cells per mm cube. Do remember. Clear? In any person, less than 200 cells per mm cube, that is TH cell count. That is an indirect evidence of HIV. Do remember this. Clear? So that in case of HIV, specialized test called as CD4 count, and one more is there, CD8. That is also cluster differentiation aid. So CD4 and CD8 count is also one of the method to diagnose the HIV. Clear? Once again, ELISA refers to enzyme linked immunosorbent assay. That is, the main important role is antigen antibody reaction. And second one is CD4 count or CD8 cell count, because in a normal healthy person the count of the TH cell is almost 500 to 1200 cells per mm cube. But in case of HIV patient, there the number or the count of the TH cell can be reduced. Reduced means less than 200. If you get the count that is less than 200 cells per mm cube, means it is an indirect evidence of HIV. This is what the diagnosis. Next. Symptoms. Yes? Question mark. Yes. Can you guess what are the symptoms for HIV? Yes. There is no, there are no specialized symptoms for the HIV. I told you HIV is a syndrome. Syndrome means, I told you, group of diseases. It means that the person who is suffering from HIV, that particular patient is prone to many infection. Means more chance to get infection, isn't it? So it means that overall I am telling you people, the person who is suffering from HIV, in their body there is no immune system. I told you there is no immune system means he is more prone to get disease or infection. So that in their particular body, that is in HIV patient body, there is no immune system. So he may start suffering from typhoid, TB, even common cold, isn't it? Whatever the diseases. So many symptoms, many diseases, so many symptoms. Clear? One disease equal to approximately 4 to 5 diseases, symptoms. The HIV patient may suffer from, at a time he is going to suffer from many diseases. Many diseases means many symptoms. This is what? The symptoms. Next, treatment. So I hope till now there is no particular FDA approved medicines for the HIV. FDA means Food and Drug Administration. It is newer. So there is no approved medicines for the HIV. But however, our scientists have discovered some of the medications for the HIV, but not to cure. But however, we can increase the lifespan of the patient. Do remember, these all medicines, whatever we are discussing here, these all medicines can be helpful for the person or the patient to just to increase the lifespan, but not to cure 100%. Clear? So there is no such medicines for the treatment. However, we have some medicines. In that, first one AZT, that is azithromycin. So remember, AZT means azithromycin, and one more is DDI, that is dideoxyinosine. So the main role of these two medicine is to prevent the enzyme, that is reverse transcriptase. It means that these two medicines play a very important role. So imagine this is what HIV, the TH cell, that is T helper cell. So when any virus attached to this helper cell, then it is going to release its genetic material, that is single stranded RNA. As you know that, called as release. So the main step is the single stranded RNA has to convert it into double stranded DNA, called as viral DNA. Isn't it? Then only it will get an ability to cause disease in case of human beings. So now these two tablets or medications play a very important role in the prevention of conversion. I hope you understand. So if this virus is going to enter or injected into the cytoplasm of the cell, these two medicines or tablets are going to play important role. They prevent the conversion. Means these two medicines never allow the RNA into DNA, conversion of RNA into DNA, so that when this RNA never converted into DNA means it never incorporate in the DNA. It never incorporated in the DNA means there is no possibility of synthesis of viral particles. Isn't it? So that however we can prevent the load of virus. Clear? However they are going to produce, some of the viral particles are going to produce, but we can prevent the load, means number of virus can be reduced. So this is what the capacity or the ability of the medicine, that is azithromycin and dideoxyinosine. Next, these drugs can inhibit the enzyme of HIV, that is reverse transcriptase. Means these two medicines can prevent the conversion from single stranded RNA to double stranded DNA. Next, most important, highly active antiretroviral therapy called as HART. HART means highly active antiretroviral therapy, means some of the medications which is given to a person or patient against that virus, called as antiretroviral. Retro means come under HIV, anti means against. The medication which is given to against the virus, called as antiretroviral therapy, simply called as HART, is a combination of 3 or more antiviral agents. Clear? More than 2 medications are given at a time, so called as combination of 3 or more antiviral agents, called as triple therapy. Important word, called as triple therapy or HART, which has been highly effective in reducing the number of HIV particles in the bloodstream. So whatever the number of viral particles are present in the cell, after that they are going to break open the cell and comes out of the cell and enter into the bloodstream. So there, this medication is going to prevent the number of viral particles in the blood. So by means of all this, however, we can increase the lifespan of the patient, but it is not possible to cure that HIV 100%. It is not at all possible. Clear? So reduce number of HIV particles in the bloodstream, and as a result of increase the number of CD4 count. I told you CD4 refers to TH cells. Clear? So by means of this we can increase the number of TH cells. Next, prevention of AIDS. So already we discussed in first 10th standard, it was there. Ensuring use of disposal syringe. Clear? Disposal syringe means only one time we are going to use the syringes. Ensuring use of disposal shaving blades, as we know this. Screen the blood before transfusing from donor to recipient. Clear? So it may be emergency or it may be so whatever the thing. So that time the doctors has to screen the blood before transfusion. Here the word screening refers to before transfusing from donor to recipient first we need to check the status of the donor's blood. Here the word status refers to, we need to understand to which disease he is suffering from. So the donor may suffer from HIV, the donor may suffering from a TB and other diseases also. So for that reason, before transfusing, first we need to screen the blood of the donor. Advocating safe sex means use condoms. So they are considered as mechanical barriers. So they can avoid STD, that is sexually transmitting diseases. Then avoid multiple sex. Then from mother to her respective child. Then there are some agencies. NACO refers to National AIDS Control Organization. National AIDS Control Organization. And many NGOs are doing a lot to create awareness. So among the people, it means that being educated people, being what the neighbors or your child or your friends, whoever it may be. So as much as possible, try to give awareness about the AIDS. Clear? So what are the consequences or what are the problem with the AIDS? We need to give awareness to our neighbors, child or people or whatever. So WHO has started number of programs to prevent the spreading of HIV. So December 31st, as you know that. So we are going to celebrate World AIDS Day, isn't it? So free distribution of condoms and controlling drug abuse. There the drug abuse refers to misuse of drugs. Clear? And regular checkup for HIV in susceptible population. Next, cancer will discuss in the next class. Thank you very much.